grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maraj and this lesson is brought to you by the Gauteng Department of Education in collaboration with Cybono Discovery Center. We also have a WhatsApp line for the lesson, so if you have any questions, you can please WhatsApp to 079 454 9535. That's 079 454 9535. Please do not send voice notes or pictures or photos. You can type out your questions and then we will get back to you. Right, so today's lesson is going to be on meiosis part 2. Right, so last week you did meiosis part 1, so now we are moving to the second part of meiosis, and the lesson today is going to be by myself, Vedika Maraj, and my co-presenter is going to be Sina Sekeke. So let's just go have a recap on meiosis that you did last week. We're not going to go through all the phases, but we just want to remind you what a meiosis is. And it is a cell division whereby a diploid cell undergoes 2 cell divisions and divide to form 4 different haploid cells. Okay, so if you can think about it, essentially a diploid cell is a somatic cell. So we are saying that that cell is going to go through 2 divisions, and then in the end we are going to result in 4 cells, and those are going to be haploid, and those are essentially our gametes. So as the picture shows over there, it shows you clearly the 2N. It even shows you DNA replication. And just to remind us, which phase does DNA, when does DNA replication take place? During interphase. And interphase is known as the preparatory phase, and it is the phase between cell cycles. Okay, and then it shows us that we have meiosis 1. So that is when we're going to get from the 2N to the 1N. That is when the number is going to be halved, and then we're going to have meiosis 2, where the number is now maintained. And if you remember correctly, this is also the phase 2, that is similar to mitosis. Mitosis is a different process. It's also cell division, but that one is the one that you learned about in grade 10, and it doesn't give rise to haploid cells. Okay, it's going to have the same number of chromosomes as we have in the beginning. Right, so that is basically meiosis in a nutshell. So you need to understand this flow diagram in order to understand what we are going to discuss today. But before we get on to that, let's also remind ourselves what the importance of meiosis is. So there are 3 main things. Number 1, the production of gametes, which I already spoke about, which we said is 4 daughter cells that are formed. Those are the gametes. And then number 2, halving the chromosome number. So going from the diploid to the haploid. So the chromosome number remains constant from generation to generation within a species. Okay, so why is this important? We need to keep a specific species is going to have a specific number of chromosomes that's going to make that organism, that specific organism, right? So if we don't halve the number, we are then going to be resulting in, if we don't halve the number for a gamete, when 2 gametes fuse, we're going to be having too many chromosomes and we're not going to be having that organism anymore. So it's important that we halve the number so that we have now gametes that have half the number, so that when gametes fuse, we have the full complement again, the full set, which is the diploid. And then that organism will be the organism it's supposed to be. Then lastly, the mechanism to introduce genetic variation. Very important. It's a mechanism to introduce genetic variation. How does it do it? Genetic variation meaning that it's all going to be different. They're going to be different because the 4 cells that are the 4 gametes that are formed at the end of meiosis are going to be genetically different. And this is brought about by 2 things during meiosis. Number 1, crossing over during prophase 1, because if you can remember correctly, though, that is when homologous chromosomes are going to lie adjacent to each other and chromatids are going to overlap. It's the point called the chiasma, and they are going to exchange genetic material. So those chromosomes are now going to be different, right? So that is going to cause genetic variation. And then the other one is the random arrangement of chromosomes at the equator during metaphase 1 and metaphase 2. Right, so now we move on to the business of today. And let's look at the examination guidelines. So in total, paper 2, meiosis falls under paper 2, and it's 21 marks, which forms about 14% of the paper. But today we are going to be talking about abnormal meiosis and the comparison of mitosis and meiosis. Grade 12s, I want to remind you that when you are studying or when you are doing this topic, you must ensure that you understand and are able to answer questions on every bullet that is in the examination guideline. Okay, so number 1, it says there, non-disjunction and its consequences. Right? Number 2, non-disjunction of chromosomes at position 21 during anaphase in humans to form abnormal gametes.
You have been given power to decide whether you want good results in your life, in your schoolwork, or you want bad results. As Tony Robbins said, it is in the moment of your decision that your destiny is shaped. And your destiny is shaped right now with the decisions that you are currently making in your schoolwork. So decide whether you want to be an A student and start working on it, because there is power inside of you. The infinite power is inside of you, and the time you decide to tap in that power, you will become who you want to become in your schoolwork.
Let's just go back to the examination guideline again. What does it say about meiosis, and what do you need to know about meiosis? So it's in paper 2, and it's 21 marks, and it counts about 14% of paper 2. Okay, now I want to remind you, grade 12s, with regards to the examination guideline, this is a very important document, and what you need to basically look at is you need to make sure that when you are studying or learning this topic, that you are able to understand and do activities on every bullet that is in your examination guideline. So today we are going to be looking at abnormal meiosis and comparison of mitosis and meiosis. Now remember, I mentioned to you that mitosis is the one that's from grade 10, right? So the first bullet says you need to know about non-disjunction and its consequences. Number 2, non-disjunction of chromosomes at position 21 during anaphase in humans to form abnormal gametes with an extra chromosome of a copy of chromosome 21. Then the third bullet there, the fusion between abnormal gamete 24 chromosomes and a normal gamete 23 chromosomes, which may lead to Down syndrome. Okay, and then the last 2 is similarities of mitosis and meiosis and the differences between mitosis and meiosis. Okay, so we're going to do it in more detail now, but this is just to show you what it is that you need to know based on the examination guideline. Right, now we go the abnormal meiosis. Now what I'm talking about here, what does it mean when we talk about abnormal meiosis? So what we're talking about here is we are talking about chromosomal mutations that can occur. Chromosomal mutation, okay, and you'll see some sources also call it aberration. Okay, so that is when we have an abnormality occurring where we have an extra chromosome, or basically it's a change in the number of chromosomes. It's in the number or the shape, okay, but it's an alteration that occurs in the chromosome. So it's like, for example, having an extra chromosome or having less chromosomes. Okay, so that's a changing in the number of chromosomes, and that leads to an abnormal meiosis because now because we are not going to have the chromosome number maintained the way that we want it to. So that's the abnormality. Okay, now what causes this chromosomal mutation? How is it possible that at the end of meiosis we are going to have either extra chromosomes or we're going to have less chromosomes? And this is due to a process called non-disjunction. Okay, it's a process called non-disjunction. This process is what's going to cause the chromosomal mutation. So the failure of chromosomes to separate during meiosis, that is what non-disjunction is. The failure of chromosomes to separate during meiosis. Now, without reading there, let's think about it. At which point do chromosomes separate during meiosis? You should have said during anaphase 1 and during anaphase 2, right? So let's see what happens during anaphase 1. If we have this non-disjunction occurring at anaphase 1, this is when the homologous chromosomes are not going to separate. They fail to separate. Okay? The homologous chromosomes, remember, they're in pairs, they lie next to each other during metaphase 1, and if they fail to separate during anaphase 1, that is one of the ways, right? The second one is if during anaphase 2, the chromatids fail to separate. Now let me remind you, during metaphase 2, we're going to have chromosomes that are going to lie singly at the equator. They're not in pairs. Okay, because remember, they got separated during meiosis 1. Now in meiosis 2, they are lying at the equator. Now, during anaphase 2, remember what happens? The centromere, the spindle fibers are going to contract, right? Or they're going to shorten. The centromere is going to divide or split. And the chromatids are going to be pulled towards the poles. Okay? The chromatids in anaphase 2. So it's very important. Why I'm emphasizing this is that it's important for you to know that when we are talking about anaphase 1, we need to refer to the homologous chromosomes that fail to separate. And when we are talking about anaphase 2, then we are talking about chromatids that fail to separate. Okay, recap. Non-disjunction is about the failure of the chromosomes to separate during meiosis. And it can happen during anaphase 1 when homologous chromosomes fail to separate, or anaphase 2 where the chromatids do not separate. Right, so this is now showing us what is going to be happening in a normal cell and in 2 abnormal cells. Okay, so let's go through this. I know there's no words here, but I want you to listen to what I'm saying and look at what I'm referring to. So let's look first at the normal one, okay? We're looking at, this is the one we are doing first. If you can see that cell, it's a diploid cell. I just want to put, yeah, we're going to have, this is the diploid cell, so this is going to be 2N, okay? And in this cell, we now have how many chromosomes do you see there? 4, right? You should have had 4. So at the end of meiosis, we're expecting to have how many in each cell? 2, right? So let's look at the bottom over here. What do we have over here? It's 2. So here in the normal case, we have N, which is going to be at the end of meiosis. And even here, we also have N. Do you see that? Okay, because why am I saying we have N here? Because after the first meiotic division, after meiosis 1, we are now halving it. We had 4. We had 1 cell with 4 chromosomes. Then after meiosis 1, we now have 2 cells with 2 chromosomes each. Then each of those cells are going to go through, meiosis 2 is going to happen, and then each of those cells are going to further divide into another 2. And that's how we land up with 4. Okay, and then that number of N is maintained. You can see that we have 2 chromosomes here, and now we also have 2 chromosomes here in each cell. Fine, normal meiosis, you all understand that. Now we move on to the abnormal one. This is when there's a problem in meiosis 1. This little star box, you can say, like, it looks like a little firework, that is showing us that the problem occurred there during meiosis 1. So let's look once again at the first one. This is 2N. Okay, so what's going to happen here is, now see, something went on over here in meiosis 1. During anaphase 1, the homologous chromosomes failed to separate. So you can see there's a homologous pair here and there's a homologous pair here. So one of them did not separate. So it's actually the first one that they're showing there. It did not separate. So you can see that in this cell now, we have an extra chromosome. Because what was supposed to happen was, we were supposed to have 2 in this cell and we're supposed to have 2 in this cell. But now they did not separate. So it stayed on the one side. Okay, so it's going to form part of the one cell and the other cell is going to form. And the other cell is going to be having 1 less chromosome. Okay, so during anaphase 1, homologous chromosomes failed to separate, which resulted now in this, at telophase a telophase 1, where we're going to have one cell with 3 chromosomes and the other cell with 1 chromosome. Now, this is still going to cause a problem now in meiosis 2. It's not like the problem is only in meiosis 1 and now it's going to be solved in meiosis 2. The problem is going to continue. Why? Because each of those cells need to divide, right? And the chromosomes need to split. So the chromatids need to move apart during anaphase 2. So what's going to happen here is that this cell, the chromosome here is going to divide in this one, and it's going to go into 1 and 1, and then this one, they are also all going to divide, and that's how we're going to get 3 and 3. So do you see that in this instance all of them are affected? So if the mutation or the non-disjunction occurs in anaphase 1, that is going to result at the end of meiosis 2, all the cells are going to be abnormal, because we are going to have 2 cells that have 1 less chromosome, and we're going to have 2 cells that have 1 extra chromosome. You see, so what we can basically say over here is that this one is n minus 1, this one is also n minus 1, and this one is n plus 1, and that one is n plus 1. So what do I mean by that? I mean that if this was a human, right, they're supposed to have 23 chromosomes at the end, which is n. So here we all have n. Each one of these is n in the normal case. So in this one, if we have 1 less, so we have n minus 1, and in this instance we have n plus 1. I'll tell you now why I'm teaching it to you in that way. Now we move on to abnormal, the other one now. Okay, this is when we have non-disjunction that is occurring in anaphase 2. So here, once again, we start with our 4 chromosomes, right? And then during meiosis 1, they're going to separate, and we're going to get 2 chromosomes. So here we have N, and then here we have N. Everything is perfect. Now we come to meiosis 2. Now, all of a sudden, during anaphase 2, our chromatids are going to fail to separate. Okay, and now what is that going to result in? In this instance now, it's going to result in 2 cells having N, right? 1 cell having N minus 1, and 1 cell having N plus 1. So here we have 2 abnormal cells, and we have 2 normal cells. Now, the reason why I'm explaining to you about the n minus 1 and n plus 1 is because the examiners aren't always going to give you a question on humans. Okay, so if it's on humans, if they are humans, we are talking about a human being, then our 2n equals to 46 chromosomes, and our n equals to 23 chromosomes, right? So I could easily have rather said that here we have 22, okay, and here we have 24. However, the examiner might ask you a different one. They might talk to you about a pineapple, or about a cat, or about a dog. So if you just understand that we're going, when we have n, then we know that whatever they give you, the context of the question is important. So they will tell you how many chromosomes the pineapple has, for example, or the dog has. And then you'll need to work out, they'll either tell you what the diploid number is or what the haploid number is. And then you can apply that. So if the haploid number for a dog is, say, for example, 20, so in the end, if it's n minus 1, then you must know that then that one will have 19. And if it's n plus 1, then it will have 21. Okay. But that is for application when you start doing questions. Now we go to another representation. It's basically what I was explaining to you, but now it's just divided into anaphase 1 and anaphase 2. So let's have a look at this one. This one is telling us non-disjunction in anaphase 1. And by the way, this comes from your DBE textbook. Okay? This diagram, if you want to refer. All right, so now they're saying the non-disjunction happened at this point here. So what did we say when that happens? It's when the homologous chromosome fails to separate. So you can see here, this here, we have a homologous chromosome. It's homologous chromosomes, okay? It's a pair. And they fail to separate. So can you see that when during anaphase, they're going to go into this one cell. And this other cell, it looks like it's got nothing. But let's rather say that it's got one less. Okay? Because remember, 2 went that way, so it has 1 less. Because remember that in our diagrams, we can only essentially show you a few cells, but there are many other cells. So when we are sometimes showing you these representations, and when you get it in an exam, it's only going to show you a few cells sometimes, right? So it's rather better for us to talk about having 1 more and 1 less, compared to having nothing, even though you can see that there is, it looks like there's nothing there. Okay, so what's going to happen now is that then obviously this one is going to carry on, okay, and then it's going to carry on dividing, and then we are still going to have 1 less, and these 2 we are going to now have 1 more. So what do we say here? We say that anaphase 1, non-disjunction in anaphase 1 results in 2 cells with an extra copy of chromosome and 2 cells with 1 less chromosome. Okay, so here we have 1 less, and here we have 1 more. Now, if it's in in this instance, all cells are affected, right? But whereas it is then in anaphase 2, this results in 1 cell with 1 less chromosome and 1 cell with 1 extra chromosome and 2 cells that are normal. So here we have the 1 extra, 1 less, and the 2 normal. So if this was a human, then we would say that these are n, this is n minus 1, and this is n plus 1. Okay, so I hope this explains it, you understand this. Right, now before we get to the next, okay, yeah, let's do this is another picture now. Now this one is showing us here that it takes place in anaphase 1. Both cells are affected. It's exactly what I explained to you, but I just want to show you that this is showing us now. This is showing us, I just want all the things to pop up. There we go. So this is now showing us when it takes place in anaphase 1 and when it takes place in anaphase 2. So let's first look at this side. Anaphase 1, both cells are affected at the end of meiosis 1, and then all 4 gametes are affected at the end of meiosis 2. So you can see over here that all of them are affected. When it takes place during anaphase 2, then only 2 gametes are going to be affected. Okay, because as you can see how it happens here, what's nice about this diagram is that it shows you separately what's going on. Okay, so actually let's go through this a little bit more detail. So what they're showing us over here in anaphase 2, so here we see during meiosis 1, it's separated normally. We started with 2 chromosomes, and now we have 1 in either cell. Now during anaphase 2, now can you see what's going to happen? In this one is normal. This one is normal. Anaphase 1 is occurring normal. There's no non-disjunction here, and that's how we're going to land up having a separate there. But in this one, we're going to have a problem here, right? So that one's going to fail to separate, so then we're going to have this one with an extra 1 and this one with 1 less. Okay, so it doesn't just stop there, because now something must happen. How does an organism land up having the extra chromosome? That is the important part where a lot of learners miss out on. So let me repeat that. Now we spoke about abnormal gametes. Okay, you know that we have gametes with extra chromosomes, we have gametes with less chromosomes, but how does this result in, how does it affect an organism? How does it affect the zygote? And how it's going to do that is that when these affected gametes fuse with normal gametes, the zygotes will either have 1 less chromosome or 1 extra chromosome. Okay, so what does this mean? Let me give you a simple example. These are now showing us that if we have let's say it's an ovum, okay? Let's say it's an ovum, and it's going to have, say it's a human, and it's going to have 24 chromosomes, right? So you should know that in a human, an ovum should have 23. So if it has 24, it's got an extra chromosome, right? Now we add it to a sperm, and this sperm has 23. This is a normal sperm, okay? It's not an abnormal gamete. 23 is normal. That's how many we're supposed to have. So if you have that, it equals to 47 chromosomes, for example. But there's 2 things that you need to realize. Okay, so then if it's 47 chromosomes, remember that a human is supposed to have 46. That is normal. So if it's having 47, it is causing a disorder. Before we get to that, I want us to talk about what disorders will happen. I first want us to understand that it's not always happening in humans. Okay, non-disjunction doesn't only happen in humans. I spoke to you about it earlier. So here you must be able to say what you need to understand is that when an affected gamete is going to fuse with a normal gamete, that is when you are going to have either one more chromosome or one less chromosome. Okay, so that's how the zygote lands up having the extra or the less. That's what I want you to understand. Okay, but now we are moving on to what happens in Down syndrome. Okay, so what happens? So non-disjunction, one of the things that non-disjunction can result in is Down syndrome. Okay, it's just one of the disorders that non-disjunction can cause. And this is the one that you need to know in your syllabus. According to the examination guideline, you need to know about Down syndrome and about how Down syndrome occurs. Okay. So in how we explain it is that, firstly, before I explain it with the words according to a marking guideline, I first want to help you to understand it, right? Let's look at the picture on the right-hand side. So this is showing us that if we have a problem in meiosis 1, so we have a mother cell having 46 chromosomes, right? That is now 2n. It's diploid. And then meiosis 1 occurs and will result having an ovum. It's not an ovum, we're going to have a cell with one more chromosome, and we're going to have one with one less. And then when they divide, we are still going to have one with one more with an extra chromosome. That's an ovum. Okay. When the sperm cell is going to come, which has 23, which is normal, then we are going to have a zygote with 47. Okay. So that's what I explained to you earlier as well. There's just a different representation. However, there's two things that we want you to first understand about non-disjunction that results in Down syndrome. Firstly, it can either take place in anaphase 1 or in anaphase 2. Secondly, it's not always the ovum that is going to result in an extra chromosome. It can also be a sperm cell. Okay, it's more often, in majority of instances, it's the ovum that gets the extra chromosome, and in majority of instances, it is Down syndrome is the non-disjunction is occurring in anaphase 1. However, it can happen in both. So when you are when you are answering a question on Down syndrome or anything in your question paper, you need to be able to read the information that is given to you in the question. So the context of the question is very important because it's going to tell you whether we're talking about a human, which chromosome pairs are affected, because the other important thing about Down syndrome is that the separation occurs on chromosome pair 21. So we have in our human somatic cell, we have 22 pairs of autosomes and 1 pair of gonosomes. Okay? And on the 21st pair, if there's failure of separation or there's non-disjunction that affects the 21st pair, that is when it's going to now result in the extra chromosome, right? And it's going to be the extra copy at that chromosome pair. So that is about non-disjunction. So let's look at the answer. Okay, but then after that, now we have a gamete, as I explained, with one extra chromosome. It's got an extra copy of chromosome pair 21. Okay, so now there are 3 there. In a that is in the individual, right? But remember that the non-disjunction is going to occur during meiosis. So if it's going to occur during meiosis, then at the end we are going to have a gamete that is going to have 1 extra chromosome, right? So in that karyotype, you are going to have 22 autosomes, and you're going to have 1 gonosome, because remember it's a haploid cell, right? So in that instance, on pair number 21, there's going to be an extra copy. That's where we are. Now, how does Down syndrome happen? That is when we have a gamete, that abnormal gamete that is going to have an extra chromosome. It's going to fuse, wait, let me back that up again. Let me repeat what I said about the karyotype of a haploid. If it's going to be chromosome 21 that does not separate, now we are not going to have 23, we're going to have 23 autosomes, right? And we're going to have one gonosome. And that is going to bring us to our 24. Okay, so that's how we're going to have a haploid cell. We're going to have a gamete with 24. So when that gamete is going to fuse with a normal gamete, which I've been saying now for the last few minutes, we are going to have a zygote with 47 chromosomes. Okay, so let's look at the explanation that we have on the left that comes from the marking guideline. In meiosis 1, anaphase 1 of chromosome pair 21 does not separate. And it can be in, or is very important there. In meiosis 2, during anaphase 2, the chromatids of chromosome 21 do not separate. So we see there, anaphase 1, we talk about chromosomes, and 21 is important. And anaphase 2, we talk about chromatids, and once again, the 21 is important. This is referred to as non-disjunction. One gamete will have an extra copy of chromosome number 21. And if this gamete fuses with a normal gamete, the resulting zygote will have 3 copies of chromosome 21 instead of 2. So that means we're going to have a karyotype of 47 and not 46. And that is going to lead to Down syndrome, which is also known as trisomy 21. Okay. So the other thing that learners get incorrect here is that they think that all non-disjunction has got to do with Down syndrome. As I explained to you in the beginning, this non-disjunction can happen in animals, it can happen in plants, it not necessarily happens only in humans. But if it happens in humans and it affects chromosome pair 21, and those abnormal gametes are going to fuse with a normal gamete, that is when we are going to have Down syndrome. So these are the karyotypes I was talking about, okay? So these are ones of normal people. This one is a male and this one is a female. So what it's showing us over here, these are the 21 pairs I was talking about, the pairs, the 22 pairs, right? So it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Okay, and then the 23 pair is the gonosome. So this is then the 21. So if it is Down syndrome, then there's going to be an extra 1 here. Okay, this is now a diploid organism. As you can see, there are 23 pairs. So in this one, there will be an extra copy. But the haploid cell that was having an extra chromosome is going to have not 3, it's only going to have 2. Now, how is it going to look if somebody has Down syndrome? It's going to look like this. So in a diploid cell, this is now in 2N, where we've got 22 pairs of chromosomes, and we have 1 pair of gonosomes, 22 pairs of autosomes, and 1 pair of gonosomes. You can see here at number 21, we have 3 copies of number 21. And that is because an abnormal gamete with an extra chromosome fused with a normal gamete, and that's how we came to 47. Okay, and this is if the individual has Down syndrome or trisomy 21. So these are some of the symptoms that somebody with Down syndrome would have. Okay, these are the features that they have. And then we're moving on to our differences between mitosis and meiosis. Okay, so the main thing is that in mitosis, it occurs in body cells, and in meiosis, it occurs in the sex organs. Okay, and in mitosis, there's 1 division that occurs, and in meiosis, there are 2 divisions. In mitosis, there are 2 daughter cells that are formed, and in meiosis, there are 4 daughter cells. In mitosis, the daughter cells are identical to the mother cell. They're identical to each other and to the mother cell. And in meiosis, the daughter cells are genetically different from each other and from the mother cell. That's why we have genetic variation there. And then in mitosis, we have chromosome number remains constant, and in meiosis, the chromosome number is halved, and in mitosis, crossing over does not occur, and in meiosis, crossing over occurs. Okay, so those are the ones that you need to know. Okay, and yeah. Then the similarities between mitosis and meiosis. Both processes are cell division processes. Both require DNA replication to occur during interphase before the division phases begin, and both show the following phases: prophase, metaphase, anaphase, and telophase. Okay, I just want to quickly go back. Give me a second. Okay, we can stop on this one. I just want to reiterate something. You need to know non-disjunction, okay, as we explained. And you need to know that non-disjunction is going to result in abnormal gametes, right? So they're either going to have extra chromosomes or they're going to have less chromosomes. And so non-disjunction, as I said, it can happen in animals, it can happen in plants. It doesn't always happen only in humans. But when it happens to humans and it affects chromosome pair 21, that is when we are having that results in Down syndrome. Okay, that's the first point. Then the second point is that when you are doing questions, and I'm sure Sina is going to talk about this also in length, but when you are doing questions or when you are doing activities in class, make sure that you read what is the question giving us. What is the question telling you about what is happening? Because when you need to talk about the chromosome numbers and you need to talk about abnormal gametes or how many chromosomes are going to be resulting, you need to know what are we looking at. What learners tend to do is they apply non-disjunction to Down syndrome for everything. Every time they see a non-disjunction question, they think it's Down syndrome. Every time they see non-disjunction, they want to talk about 47 chromosomes and trisomy 21. But sometimes the examiner is giving you a question on non-disjunction that has nothing to do with Down syndrome. Okay, so it's very important that you read the questions very carefully, and also more importantly, you understand why is it happening in anaphase 1 and what is the result, and what's happening in anaphase 2 and what is the result. What I find the easiest thing to do in this instance is that you draw the pictures. Draw the pictures. Start with 4 chromosomes, like I showed you in the first diagram that we did. Draw the chromosomes, separate them. Think to yourself, draw them and see how they're not going to separate, so how is it going to result? When you do drawings in life sciences, they really help you to understand difficult concepts. Okay, so that is all from my side. We're going to quickly have a quick 5-minute break, and then I'm going to hand over to my colleague and my co-presenter, Sina, that's going to take you through some questions and activities on the abnormal meiosis.
grade 12s. My name is Sina Sekheche, and I would like to please pass my thank you to my co-partner, Vedika Baharaj, for setting up the tone for today. Well, I hope you guys learned a lot with the abnormal meiosis and non-disjunction. I know it's quite a lot, it's a lot of terminology. You just need to follow through on the diagrams and check the number of chromosomes you have in each cell. Then you will know. Then the concept is non-disjunction, what it leads to, and that is your chromosomal mutation. So please just make sure that you follow through the terminology, you get used to talking about it and using the correct one, because then you lose marks. You understand the concept, but you're not able to write it out for you to get full marks. So we are going to look at how the examiners are asking questions on these topics. Remember, these are just topics from the previous question papers or from the previous years that show you the style of questioning, but it does not mean that the questions will always appear in this way. So what does that mean to you? It means you need to study the content, know your concepts correctly, know the definitions, know the processes, so that you are able to answer any question that may be posed to you. So these questions are just possible questions that might appear or that you may come across. So then you need to be equipped in how to answer these questions. So let us look at activity 1. I know most of you, in fact all of you, are familiar with answering multiple choice in life sciences. This is our first question in the question paper, question 1.1. So let us check what this question has here for us. This will be sort of like a warm-up activity for us. Now, the question asks you, which option in the table below, now this table, shows the correct? Please make sure that you highlight. When you are reading or going through a question, you need to check what are they asking you for. So here they want the correct comparison between mitosis and meiosis. So you've done that just now. So this is the last thing that you did with Vedika. So you might remember this at the top of your head. So I would like us to go through this question together. So when you go through a multiple choice question, you use the method of elimination. You start by looking at those answers that are completely incorrect, and then you start eliminating them until you get to the most suitable answer, or you might even get 2, and then you can just check between the 2 that you have. So let us start with the first one, the first option A. It says mitosis produces 4 daughter cells. Now you sit back and you think, mitosis. This is what we did in grade 10. How can you associate yourself with mitosis? Just remember, this is the process that you did in grade 10. And in grade 12, you did not do the process in detail. You only did the comparisons between the process that you are doing in grade 12, which is meiosis, where we are forming gametes. Now, you remember now, in grade 10, when you were doing meiosis, were you talking about 4 daughter cells from one cell? Then no, then you remember then to say no, this is not correct. Then you come to meiosis, it produces 2 daughter cells. And then you think, now I have just done meiosis in grade 12. What number of cells were resulting at the end of this topic? It is not 2, definitely. Actually, this is vice versa. So you see, in meiosis, we produce 4 daughter cells. And in mitosis, we produce 2 daughter cells. We have not spoken about their genetic material or what type of genetic material they carry. We just speak about the comparison that we see. So we see now A is out. We cannot have these because they are now vice versa. Then you come to B. Produces genetically different cells. Mitosis, grade 10. Were we talking about genetically different cells? No. How were the daughter cells? They were the same, genetically the same. Then you say no. Because it says different, they were identical. Then you come to meiosis, it produces genetically identical cells. Remember what meiosis produces? It's gametes. What are those types of cells that are produced at the end of this process? They are called gametes. So can gametes have the same genetic material? Can they be genetically identical? No. Then this, because they are incorrect, you are going to eliminate B as well. Then you come to C. The chromosome number remains the same. You think about it. Okay, so this one, it is true about mitosis because the mother cell will have the same chromosome number as the daughter cells. So we can keep this one and check the other. Remember, you have to eliminate all. You don't just jump to the correct one, even if you see it. Then you come to meiosis, which is the second part here. The chromosome number is halved. Remember, Vedika was telling you about the importances or the significances of meiosis. So meiosis halves the number of chromosomes to maintain the chromosome number for particular individuals or species or organisms. Remember, she said that if these organisms, the number of chromosomes are not halved, then it means it will give us an abnormal gamete and it will not give us that particular organism, because the number of chromosomes also determines the organism that is going to be formed. So this number needs to be maintained. It needs to be halved, and that was the significance of meiosis. So this can also be true about meiosis, and then you just leave it to say, okay, question mark, I will explore this, but I want to explore all my options. Then you come to D. D, it says, in mitosis, 2 divisions occur, you think. Now, do we have mitosis 1 and mitosis 2? No, this was done in grade 10, and there is no way we have mitosis 1 and mitosis 2. So no 2 divisions are occurring. And then in meiosis, 4 divisions are occurring. Check the number, 4. It means the cell is going to divide and divide and divide and divide. So do we have that in meiosis as well? No, in meiosis we only have 2. In mitosis is just 1. So this option is not correct. So this leaves us with what? With option C. Did you see how much time we took to analyze these multiple choice questions? So I want you to think that not all multiple choice questions are easy, or you can just pick and choose without going into the question and analyzing and checking. So this might seem like a simple table, but it needed you to dissect all the possible answers and then eliminate those that are incorrect. So please, 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 when you approach a multiple choice question, make sure that you follow the steps that we've just followed for any type of multiple choice question. And it is given 2 marks for a purpose. You have 2 marks, you have 2 minutes for you to go through the entire question. So please don't just pick, make sure that you give quality answers in your multiple choice question. Then we move to activity 2. This activity, I would like for us to go through the diagram first and the scenario. What do we have here? So let us check. Sometimes an error occurs. You've spoken a lot about these errors earlier on during the first session with my co-partner Vedika. In oogenesis. Now oogenesis, if you have not done that with your teacher, it is the formation of an ovum. So this happens during meiosis. So you've already done that actually with your teacher. So sometimes an error might occur during the formation of that ovum. That error may result in an ovum which carries an extra chromosome. So you know the number of chromosomes that you must have in a normal ovum are 23, right? So Vedika has also spoken to that. But if you have an extra chromosome, you are going to have an extra 1 chromosome, meaning your ovum will have 24 chromosomes instead of 23. So that is an abnormal chromosome. It has an error that occurred. Now, part of such a process is shown in the diagram alongside. So that is below here. So let us look at this diagram. You find a diploid cell. It undergoes stage 1, and this diploid cell, remember, diploid is 2N, undergoes the first division. You see here, it becomes 2 cells. But what is happening in these cells? You see, it was the X chromosomes plus 44. So remember, a diploid cell has 46 chromosomes. Diploid is 46, is the complete. Then when it divides, this one has now X plus 23 and X plus 21. Now you see, now there is an imbalance here. So what did happen here? So in here you were expecting to get X plus 22 and X plus 22. So under normal circumstances, this was going to be X plus 22 and X plus 22. So this was under normal circumstances. But because this is not a normal situation. What happened? This cell in here ended up with having 23 chromosomes and this one has 21. Then already this tells you something. There was an error there, like they say in our statement. And then these cells will further undergo the second division. And when they undergo the second division, they will go with the same problem that was created in the first stage. So look, what is happening here? You find an ovum, remember oogenesis, you find an ovum with an X plus 23 and an X plus 23. And then with this side, on this side, ovum T, you find an ovum with X plus 21 and X plus 21. Why is it 21, 21? Because the problem was already created. They failed to separate where? In the first division. So this is just you trying to analyze the diagram, trying to analyze the information before you even go to the questions. When we say to you, take your pencil, analyze the question, study the diagram. This is what we mean. By the time you go to the questions, already you are familiar with the scenario of what is happening. Now, let us take a look at the questions, and I'm going to give you some time to go through these questions or to answer the questions. But let's read the questions quickly. Number 1 says, name stage 1 in the diagram. Stage 1, you've already spoken about that. And I was too careful not to mention that when I was going through the diagram. Then number 2, identify the error that resulted in ovum R. Then you go and check ovum R. So what was the error that took place there that resulted in this ovum having this? Then number 3, how many gonosomes are there in a diploid cell? Then number 4, if ovum S, you go check, is fertilized by a normal sperm, what will the number of chromosomes in the zygote be? Then you will write, you use the numbers there just to give us the number. Then what genetic disorder will result? So I'm going to give you 3 minutes to answer the questions based on this diagram. So it's going to be much easier because we went through the diagram together. 3 minutes for you to go through this. Right, I think answering this one was much easier after we went through this diagram together. Then also remembering what was said during the first session or the first part of this lesson. Now, name stage 1. You see, it is even written in capital letters, uppercase, to say that go and look for stage 1. Don't just write stage 1 based on what you think you know or based on previous activities. So you go and look at stage 1 in the diagram. So what is that stage? That is meiosis 1. Now, if they were asking you what phase was that, then you would say anaphase 1 of meiosis 1, because this is where now the first division was taking place, the first separation. And this separation, you can already see that it was an abnormal one. The separation failed. Another chromosome, there was an extra chromosome in this cell. You see, you have X plus 23 when it was supposed to be X plus 22. So that was a failure to separate there. Now, identify the error that resulted in ovum R. So this ovum right here, there's an error there because a normal ovum should be X plus 22. So your 22 will be your autosomes, your normal body, your normal chromosomes, and then your X will be your, I don't want to say, your X will be your sex chromosomes. Yes, let's just say sex chromosomes for now, because I want you to know the correct terminology for that. What do we refer to them as? Right, so what was the error that resulted in this ovum? What is it called? It is called non-disjunction. And remember that error started occurring where? In anaphase 1 of meiosis 1. Then how many gonosomes? Here is the term now. How many gonosomes are there in a diploid cell? So see here, you need to apply your background knowledge on terminology. What do they mean about gonosomes? What are gonosomes? Gonosomes are your sex chromosomes. And then, what is a diploid cell? A diploid cell is a cell that has 46 chromosomes, or it can be represented as 2n. Now, how many gonosomes can you find in such a cell? So you see, terminology plays a very huge role in understanding this topic as well. So you will find 2. Remember, on your gonosomes, you will have your X and Y chromosomes. Those are your X chromosomes. So if it is a female cell or a female ovum, no, not an ovum, because an ovum is N. Be careful with that one. So this one is a diploid cell. A diploid cell is the one that has already fused, like your zygote. So how many gonosomes will be there? You will have one from the mother, one from the father, and then XX or XY. So there will be 2, right? Going further with the questions, if ovum S, you go check ovum S, this one, X plus 23, is fertilized by a normal sperm, then you think, well, a normal sperm, well, how many chromosomes must be in a normal sperm? What will the number of chromosomes in this zygote be? Now, this is what we were referring to, a zygote. A zygote is now diploid. Remember, it has from the maternal and the paternal. So how many chromosomes will there be in that zygote? There will be 47. Remember, we said a normal chromosome, a normal diploid cell has 46. However, what happened here? Ovum S has an extra chromosome because there are 23 plus X, meaning there are 24. So that 24, when you add that 24 plus 23, that is how you get the 47. So this is an abnormal gamete fusing with a normal ovum. Then it will give you this 47 chromosome, the zygote with 47 chromosomes. I think that story also makes sense. Then, what genetic disorder will result? Verica has spoken so much about a genetic disorder that will result when such happens. So you just say, remember, here you just stick to what you should study according to your exam guideline. She did say that there are others, but your focus will be just within your scope. So your scope for life sciences in grade 12 now is in the exam guideline. You study non-disjunction, which happens at chromosome 21, which is Down syndrome, or trisomy 21. So we'll talk more about that when we do other questions that have those types of separation or non-separation of chromosomes. So I think it was quite a breeze going through this activity, and then it shows you what type of questions may occur or can be asked in this topic. Now we move on to the next activity. For this activity, it says, the diagram below shows the karyotype of a gamete of an individual. Very important. Now you are used to a gamete having 2 by 2 chromosome, pair 1 having 2, 2, 2. So what is important is for you to read the stem of the question. What is given here? It says a gamete. Now you know a gamete can only have one set. So this is N. Hence, you don't see 2 by 2 here on the set of chromosomes going down. I hope you know how to read a karyotype. So then you come to the question. Name the type of mutation that occurred during the production of this gamete. So this gamete, we are already told that, okay, when it was forming, there was a mutation or an error that took place. So you must tell us what type of mutation do we call that, and then what type of gamete is represented in this karyotype. So you will get your answer. I don't want to give you the clue. Now, this gamete fuses with a normal gamete using X and Y representation. Write the sex chromosomes of the offspring. So I will give you only 2 minutes. Let's say 2 minutes for this one, for you to answer these questions, because there's not a lot of waking up or waking out the answers. The answers are just right there. So I'll give you just 2 minutes. Right. I believe that you are acing these answers. So let us check our answers if you got the same answers. Right. 3.1 says now, name the type of mutation that occurred during the production of this gamete. Now, when you checked, I gave you a hint when we started to say this is a gamete and the chromosomes are not paired. They appear how? Just in single sets. You see, I'm not saying even singly, because we are not talking about metaphase, we are not talking about, and this is a karyotype. So they are appearing in single sets and not in homologous pairs. Right. So you see, but now there is somewhere where you see there are two here. So if there are two, where they're supposed to be single, what happened? It means somewhere during the formation of this gamete, there was failure to separate of chromosomes. So what do we call that failure to separate? We call it chromosomal mutation. That is where now the chromosomes fail to separate. That error that took place. Vedika explained to you that non-disjunction or abnormal meiosis is actually chromosomal mutation, because it's when the chromosomes fail to separate. So that is an error. It's something that is not supposed to happen. However, it happens, and then it is a mutation, and it will have its consequences later, which we discuss as non-disjunction and so on. Right. What type of gamete is represented in this karyotype? Now this tests your knowledge of the female and the male gametes. So how do they look like? Or what is the karyotype supposed to look like? You know that the chromosomes that tells us the type of gamete that we have is chromosome number 23. So at chromosome number 23, this is where you find your gonosomes. What are gonosomes? Sex chromosomes. And in this case, you see that there is a big X chromosome and a small X chromosome. Now, because the chromosomes in a karyotype are not represented with X and Y at chromosome number 23, they are represented with the X's. You see, all of them were represented with the X's, but now this smaller X will tell you that this is actually our Y chromosome. Why do we say it's a Y chromosome? It is shorter than the X chromosome. There is actually even a rumor or a hypothesis that is going around in the scientific field or in the scientific environment that says that the Y chromosome is even getting shorter and shorter and smaller, that it might even disappear. But that is a story for another day. What we know is that the Y chromosome is shorter than the X chromosome. So here you will see that Y is actually shorter here. So this Y is not represented with the Y, the alphabet letter Y. But because it is shorter, then you know that it represents the Y chromosome. So the X and the Y will give you what type of gamete? This will give you a sperm cell. Remember, we don't say male because the question says gamete. I know some of you might have said this is a male karyotype or male something in the answer, but because it says gamete, this is a gamete. This karyotype is of a gamete. So what is a gamete? A sex cells. What do we call male gametes? We call them sperm. So that is why your answer is a sperm and not male. Right. This gamete fuses with a normal gamete. Already you know the story in your background. This is always the story. Please, 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 you must say that this abnormal gamete fuses with a normal. That is very important to highlight, because sometimes you are given and then you say gamete fuses with a normal gamete, but what type of gamete? You must say whether it's an abnormal or a gamete with 24 chromosomes or a gamete with extra chromosome. But because this question is linking to the previous one, they can say this. So this already accommodates this type of gamete that already has this type of mutation. So all the questions were building up to one, but in your answer, sometimes or in different questions, you must be careful that you mention that an abnormal gamete fuses with a normal gamete. That story that was told to you from the exam guideline, how you are going to answer that by Vedika, please make sure that you mention that. Now, using X and Y, we're coming back to our question. Representation. Now they are telling you what to use. Write the sex chromosomes of the offspring. Normally you have X, X, U, and X, Y. But with this gamete, are you going to have the same clean like we have? Because there's a gamete with an extra chromosome and a chromosome that was not supposed to be there, you are going to say what? Our offspring is going to actually to have, I wrote now where the answer is supposed to be, so just for clarity, it's going to have XXY. You see, why do we have two X's? Because this chromosome here came with the X that was not wanted. You see, that failed to separate, and now you fuse it with a normal gamete. What is a normal gamete? A normal gamete is that one of a female, because already we said this is a male. So a female will be your XX. So if you fuse this, will be your X in this case, because it's just one gamete, will be X. Here it was just supposed to be Y, but it is XY. So what do you have when you fuse those types of gametes? You will have X, XY, or you can put it in this order, X, Y, and X. So you will still get a mark by putting it in this format. So I hope that was quite clear. You must check. Chromosome number 23 is what gives you your gonosomes. And in a gamete, you expect to find only one set of chromosome there, either an X or a Y. But if they say this is a diploid cell, you expect to find two. A diploid cell like in a zygote, you expect to find two here. When there are two, that's when you will decide whether it's a boy or a girl. If there are two X's, it will be a female. If there's an X and a Y, it will be a male. Female, male. Only in a diploid cell or in a zygote. But if they are talking about a gamete, you expect to find only one at chromosome number 23. I hope that will also help you to answer questions further with other karyotypes that you get and how to analyze. And remember, all these other chromosomes, you were told earlier that they are your autosomes, your ordinary body chromosomes. But there's a story that we talk about chromosome number 21, but we'll come to that when we see other questions. So let's go to question number 4. So question number 4, this, I hope each and every one of you will have now a better skill of answering questions on karyotypes, because we quite have a lot today. Study the diagram alongside and answer the questions that follow. So studying the diagram, remember I said to you, you go and look, okay, there are two twos. What does 2,2 mean to you? Already you see now, this is a diploid cell. Then you find the at chromosome number 23. Remember, you see this is 22, this will be your 23. What is happening? You have two also, a short one and a long one. Already you can tell the story, what is happening there. So you've studied the diagram. Then 4.1, what is the type of diagram, or what is this type of diagram called? What is represented in this diagram? Now name the syndrome that the individual is suffering from and what gave rise to this syndrome. I will give you two minutes, but I would like to do the last question and then we'll do it together as a class and just check if we understand the fusion of all these gametes when it happens. That story about the fusion of the normal and the abnormal. But for this one, you can just go through it, 2 minutes, just answer these questions quickly. Right. These ones were easy, so you were warmed up nicely with the first 3 activities. Now you are in the hang of it. I hope you are ready to answer questions on abnormal meiosis and non-disjunction. Right. What is this type of diagram called? I did say in the previous question or activity that it is called a karyotype. Please, or you can say a karyogram, but please, nothing else than that. Remember, practice your terminology so that these words, they don't confuse you during the final examination, because we see that you are writing a whole lot of things, the karyo keys. Please remember that this is a karyotype. Right. What is represented in this diagram? Now, what are these things in the karyotype? So that is what the question means. It represents the chromosomes. So the chromosomes of a full set. So here we are not told that it's of a human being or is of what, but these are chromosomes. Now name the syndrome that the individual is suffering from. Remember what happens when there is non-disjunction and then there is an extra copy of chromosome 21. You see here what is happening. I hope that all of you were able to see this. I wanted to just highlight chromosome 23, but the questions is actually on chromosome 21. So you need to read the questions carefully and they will direct you to where you are supposed to go. Not all karyotypes have problems at chromosome 23. Not all of them have problems at chromosome 21. So you need to check the questions. So this one actually wanted you to go look at chromosome number 21. You see there are two pairs, all of them. But when you get to chromosome number 21, there is an extra copy there. So what syndrome is going to result in this diagram? So it is Down syndrome or trisomy 21. So that is when there's an extra copy of chromosome at chromosome number 21. What gave rise to this syndrome? So we did say, so for it to have an extra copy, what might have happened before? Non-disjunction. So there was a failure to separate of chromosomes somewhere during anaphase 1 or anaphase 2. So please make sure that you are able to answer that. Then we come to question number 4.5. This is the one that said we will do together. Describe how this syndrome is caused. So you need to be able to play with your words here. So that whatever that you write makes sense to a person who's going to mark. Make sure that you collect marks on the correct point. I know most of you understand the story of how it came about that we have this. But you need to be able to articulate that and be able to write it. So how do we write it? You start by saying, during anaphase, the chromosome pair 21 did not separate. So here you can say during anaphase 1, anaphase 2, but just you see this is a general phase. You are not specifying that this happened during anaphase 1 or during anaphase 2, but you know that at chromosome pair 21 there was a failure to separate. Then that's where you get your mark. Then that failure to separate, what is it going to lead to? This is going to lead to one cell having 2 chromosomes number 21 instead of 1. Remember, they're supposed to have 1. But if there was failure to separate, the 2 will come together in one cell and then another cell will just have 1. So actually there will be 3 when they're supposed to just be 2, because there was this failure to separate. So one cell will have 2 chromosomes number 21 instead of just having 1. Then when this gamete fused with a sperm cell, it resulted in a zygote that had 47 chromosomes. Now this sperm cell, if it was a sperm cell that fuses with a normal gamete, if it comes with 24, remember it has 24. Why? Because there's an extra chromosome there at chromosome number 21. Instead of having 2, you have 3. Now, if it fuses with a normal sperm cell, or if it is a gamete, a female gamete, an ovum fuses with a normal sperm cell, it is going to result in a zygote that has 47 chromosomes instead of 46. Remember, we want 46. And now that will be your Down syndrome, the syndrome that you mentioned in the previous question. And then there are 3 chromosomes at chromosome number 21 instead of only 2. You see, so that is how you just articulate your answer to be able to collect the marks. Just check where the ticks are for you to be able, and also check the previous slide that had a yellow coloring, I think, where it was explained from the exam guideline how you are going to answer this type of question, to say how did it bring to Down syndrome. Then activity 5, this diagram represents the distribution of chromosome pair 21. Now already your ears, you must be highlighted, chromosome 21, as it appears in gametes at the end of meiosis 2 in a human male. Very important. This information gives you how many numbers of chromosomes you can write, because then if it says human, you know you are working with 46, 23, then 27, 20, 26, then you will see. I mean 47, 46, yes. Right, so you see what is happening here in cell A, there are chromosomes 2, there's cell B, there are 2. Remember the diagram that Vedika showed you at the end of meiosis 2, anaphase 2, where there was failure to separate of chromatids. The last stage, where you find the four gametes that are formed, and then you found 2 that seemed like they had nothing. But she did highlight to you that it does not mean there is actually nothing. It means there is 1 less of the chromosomes that were supposed to be in that particular cell, because it failed to separate during anaphase in the previous stage. So I think that also makes sense. So here you can already see that there are 2 when they're supposed to be 1, 1, 1. So there was some failure to separate there. Now explain why the diagram represented by C and D, these 2, do not have any chromosomes. So why are they represented to not have any chromosomes? So this was due to non-disjunction. So you get a mark for saying that, or non-separation of chromosome pair. You can write pair 21 because you are already given in the stem. So you can write chromosome pair 21 because it is there in the stem. Then during anaphase 1, you know this non-disjunction happens when? During anaphase 1. Now the 2 chromosomes move to 1 pole. The problem starts in anaphase 1. So this chromosomes in anaphase 1, they were supposed to move to opposite poles, but however, due to non-disjunction, they all move to 1 pole. And if they've moved to 1 pole, the cell that is going to form there is going to have an extra chromosome, and when it divides further, it will have those extras in each cell and leaving other cells with none. So none moved to the other pole. So that is the situation that took place there. Then if gamete A, so this one here, is involved in fertilization, describe how this may result in Down syndrome. So this is a repetition. You see, they can bring it from all directions. So you see now there's an extra chromosome there. You say it's the same story. Gamete A will have 24 chromosomes. Why will it have 24? Because there's an extra one there. So you see, or you can say it will have an extra chromosome. You can still get a mark for that, for saying that. So it's either saying there is 24, or you can just say there's extra. Then, but because you were told that this is a human male, you are protected, or you are safe to say there are 24 chromosomes. Then when this gamete fertilizes with a normal ovum, you see, just don't say an ovum, say a normal one, or a gamete with 23 chromosomes. This is a normal situation. The zygote will have 3 chromosomes at position 21 or 47 chromosomes, leading to what? To Down syndrome. You know, for as long as you say 3 chromosomes at position 21, that is trisomy 21, and it leads to Down syndrome. So already this information, I think now you know it at the back of your head. You can even say it while you are sleeping. Then we can move to the last activity. The diagram below represents part of an abnormal human karyotype. Human karyotype. So this is not a gamete. A human is diploid. So you know, human diploid. So that is how you're going to start off your question. And then this is a karyotype and it's abnormal. So you don't expect things to be fine here. So you check Chromosome number 21, there are 3. Already you know. Remember we said in a diploid cell, how many sets must be at each position? There must be 2, 2, 2, 2, 2. Then at chromosome 22, there are 2. Then at chromosome 23, there are 2 as well. So you see, because it's a diploid, there are 2 here. Remember I explained the story that at chromosome, if it is a diploid cell, it's a human, you expect to find 2 even at chromosome 23. Then a male will be XY and a female will be XX. So that is the story. Now, how many autosomes are shown in this diagram? The key is here. Autosomes. What are autosomes? Ordinary chromosomes, or they are just all other chromosomes except the sex chromosomes. So how many are they? 1, 2, 3, 4, 5. Let's see. We have 5. Name the type of chromosome represented by pair 23. Gonosomes. These are our sex chromosomes. Name the disorder represented in the diagram. Now this is just a breeze, a walk in the park for you. Down syndrome. Why? See there are 3 sets of chromosomes at chromosome pair number 21. Process during which anaphase of meiosis that resulted in abnormal number of chromosomes in this karyotype. So what is that process that results in abnormal number? It is non-disjunction. Please use the correct terminology. Don't say failure to separate. That is not a terminology or a correct term. So the correct term to use is non-disjunction. Now state the gender of this person. You see, if you master this one, the gender, you must be very proud of yourself. So the gender here will be male. Why? Because you see the X chromosome is shorter than, the Y chromosome is shorter than the X chromosome. And remember we said they are not represented in XY, but the length will definitely tell you. Right. So we've come to the end of today's lesson. I would like to thank you so much for watching. We will see you again next time.